Throughout the presentation, I will talk about biofuel statistics in Brazil, NMR characterization of biofuel and it is blends, biodiesel, and it is blends with fossil fuel. By biodiesel, I mean fat acid methyl esters, average chain length from C14 to C18, NMR characterization of green diesel. By green diesel, I mean hydrogenated vegetable oil. As an important note, I should mention that the central idea was to use NMR as a characterization technique to improve the industrial process rather than performing specification tests. According to the Brazilian energy research company, renewables in Brazil accounts for 50% on last year. Sugarcane biomass was the main source. Hydropower electricity was important. And on the last 10 years, it has been a growth in the production of biodiesel and renewable and green diesel. Non-renewables accounts mainly for oil and oil products and natural gas. According to the Brazilian energy company, about 65% of last year country's energy consumption came from cargo, passenger transports, and industrial sector. In this scenario, from 2023 to 2024, fossil diesel, gasoline, and natural gas consumption decreased, biodiesel and bioethanol increased, and jet fuel remained the same. The Brazilian Agency of Petroleum and Biofuels instituted a law that requires the use of 15% volume of biodiesel in fossil diesel from last year and 20% from 2030. In a similar way, 30% of bioethanol in gasoline from last year and 35% for 2030. Soybean oil was the main source for biodiesel production last year in Brazil, account for about 69%. Corn used as a raw material for bioethanol production last year accounts for about 20%, sugarcane for about 8%, and just 2% of bioethanol came from second generation growth. The vast majority of the biodiesel production in Brazil came from the transesterification of vegetable oils and animal fats with methanol in the presence of a basic catalyst. Another possible route is the esterification of free fat acids. The major contaminants from the transesterification process are the unreacted methanol, unreacted triglycerides, mono and diglycerides, and free fat acid. In this slide, you can see a proton NMR spectra recorded at 400 megahertz of a castor oil sample. It is possible to see that we can obtain a lot of useful structural information in this non-edible oil. For example, signal with letter D corresponds to the castor oil major component. Signal with letter L corresponds to amino component, the linolenic acid. Signals with letter B and C corresponds to the all constituents of the sample, the bond, the glycerol, and the triglycerides. In this table, we can see that lots of useful information can be obtained from one single proton NMR spectra recorded at 400 megahertz or higher from one drop of vegetable oil. I can guarantee you that the same level of information can be obtained from the corresponding biodiesel sample. Actually, even more information can be obtained. For example, the estimation of average chain length, the estimation of iodine index, and other parameters. Okay, now I'm going to show you a few examples illustrating how useful is the NMR technique in order to uh, adjust the industrial process for biodiesel production. Before Petrobras decided whether it would be possible to launch biodiesel from castor oil, lots of tests were conducted in some vehicles using 20% mixture of biodiesel in fossil diesel. Here in this spectrum, we can see that biodiesel from castor oil, the main component of biodiesel from castor oil is the ricinoleic acid methyl ester. This slide shows a photograph of the interior of a diesel engine after 10,000 kilometers using 20% mixture of castor oil in fossil diesel. The proton NMR spectrum is also shown. After careful integration and careful interpretation of the NMR signals, it is possible to determine that the major constituent of this precipitate, the methyl ester of the dihydroxynoleic acid, that is a minor constituent of the biodiesel from castor oil. In the constitution of this precipitate, we can also see as a minor constituent the ricinoleic acid methyl ester. And this was a turning point in Petrobras, leading to the decision to do not produce or market biodiesel from castor oil on a large scale. Another fact preventing the production of castor oil biodiesel is the unexpected appearance of some signals in the NMR spectrum after transesterification of castor oil due to undesired side reaction of the hydroxyl group, leading to the formation of some oligomers. And the presence of these oligomers can be proved from proton 13 NMR spectra. Here you can see some useful conversion spectrum. On a side note, also, castor oil should not be the first choice for biodiesel production. It is possible to modulate the reaction conditions so that a major amount of this oligomer is produced, and this oligomer can meet specification as a biolubricant. Careful integration of some signals from the proton spectra can determine the average chain length of the oligomer produced. With the goal of finding other sources of raw materials for biodiesel production and green diesel production that I'll discuss later, Petrobras has tested a lot of vegetable oils. I will show some examples on the next few slides. The palm tree produced up to 30 times more oil than other oil seeds, and this vegetable oil is available in Brazil. But in some cases, there is a problem of excessive sedimentation preventing the corresponding biofuel from complying with the requirements on filterability and contamination. The sediment investigation by NMR proved the presence of sterile glucoside. In some cases, there is a problem of excessive wax formation in biodiesel for sunflower oil, preventing the corresponding biodiesel from meet requirements on contamination and filterability. By analyzing the proton NMR spectra of wax-free sunflower oil, wax extracted from sunflower oil, and wax extracted from sunflower biodiesel, it is possible to determine the wax constitution. In this, the wax molecules are the main esters of fat acids with fat alcohols, having average chain length of 44 carbon atoms. For those who work in a company that needs to analyze the composition of hundreds of partially converted biodiesel samples in a short period of time, and usually from mixed feeds, I recommend to perform carbon-13 NMR spectrum with the investigated decoupling mode 400 megahertz for proton, or even higher with a sample changer. It is faster and more reliable for partially unconverted biodiesel sample. It makes daily work more easy, and if it is possible to use a 10 millimeters probe or a cryo probe, will be even better. Waste glycerol is the main byproduct in the biodiesel industry from the transesterification route. The purification of the crude glycerol is a high-cost process, and it is important to find other consumption routes for this waste as generated in the biodiesel industry. Here you can see the carbon-13 NMR spectrum for three reactions for the waste glycerol conversion to valuable chemicals. Some molecules have two identical asymmetric carbons. This means that they exist as a pair of enantiomers and one diacetyl isomer, and the latter can be distinguished from the former from a carbon-13 NMR spectrum, even in an achiral solvent. As you can see, some carbons just as a neighbor of the asymmetric carbons, and potentially each isomer will have different applications. After integration and interpretation of the proton NMR spectra of a carbon-13 NMR recorded at using the investigate decoupling mode and an appropriate pulse delay, there is no great difficulty in determination the molar mass, the molar and the mass composition of the organic molecules for the production of biodiesel and waste glycerol converted to valuable chemicals. Since last year, all bunker fuel sold in Brazil has to contain 24% of biodiesel on its composition. There is an equation for the determination of the calorific value of the mixture. This equation needs the accurate measurement of the total hydrogen content of the mixture. This can be measured by time domain NMR in a benchtop equipment, 20 megahertz for potency frequency, used in a similar way as recorded in ASTM D7171 using a 18 absolute probe, 18 millimeters absolute probe, and a FID pulse sequencing. Another important point is that all bunker fuel, Petrobras has to guarantee that all bunker fuel sold throughout national territory have 24% of biodiesel in volume. For over 20 years, Petrobras has measured the amount of biodiesel in fossil diesel by infrared spectroscopy. But for some reason, the infrared method is not so accurate in the case of bunker fuel. So Petrobras NMR team developed a method to calculate the amount of biodiesel in bunker fuel using a cryogenic-free benchtop NMR equipment, 60 megahertz for proton frequency. SEMP's NMR team, in collaboration with Professor Wanderson's Romão Group at Federal Institute of Espírito Santo, Brazil, developed an application for the rapid determination of biodiesel oxidation status from the proton high-field NMR spectra. This application determines in few minutes, whether the biodiesel is highly oxidized, partially oxidized, or not oxidized, according to the appearance of some specific signals in the high-field proton spectrum. Currently, we are committed to improving this application so that a time domain NMR data obtained in a benchtop 20 megahertz proton frequency equipment will provide us with the same information. We are working with biodiesel from different feedstocks. The main idea is that a not oxidized or a partially oxidized biodiesel sample, black line, relax more quickly than a not oxidized biodiesel sample, red line, due to the appearance of some rigid domains on the molecules during the oxidation process, replacing the existing double bonds. Possibly on a near future, I will give you some good news on this topic. Green diesel in Petrobras is called the renewable diesel. It is a hydrogenated vegetable oil. Unlike biodiesel, green diesel does not require a dedicated industrial unit. Green diesel can be produced in the existing hydrotreating units. Here we can see a simplified representation of petroleum refinery with atomic distillation, vacuum distillation, delayed coking, fluid catalytic cracking, and hydrotreating unit. In order to produce green diesel, we need to add the vegetable oil or the animal fat just before the existing hydrotreating units. The green diesel produces the molecules that are produced are N-paraffins. Since the early 1980s, the petroleum industry determines the average molecular parameters in petroleum fractions combining proton and carbon-13 high-field NMR spectra. Here we can see carbon-13 NMR spectra from a feed, a hydrorefining feed, and a hydrorefining product for a fossil fuel production. We can see that after the process, there are some spectral changes. By careful integration of NMR spectra, we can obtain useful structural information. In a similar way, if we have a mixture of vegetable oil or animal fat with petroleum refining feed after the treatment, we can see some spectral change, the disappearance of the vegetable oil signals or animal fat signals and other chains. And by careful integration of the spectrum, will give us some useful structural information as well. By analyzing the average molecular parameters of the feed and the product for a mixture of fossil plus a renewable fuel production, it is clear that the hydrogen content decrease, the olefinic hydrogen decrease, and the average chain length increase, and NMR signals arise from the vegetable oil or animal fat clearly disappears after the process as expected. In this slide, we can see phosphorus 31 magic young spinning as a tool to evaluate the effect of soybean oil on hydrotreating catalyst at SEMP's pilot plant. In order to try to diminish the costs of production, several raw materials were tested, include vegetable oils with different grades of pretreatment. This study focused on the use of the gummed soybean oil to produce green diesel at pilot plant. The gumming consists on the removal of parts of phospholipids, mainly lecithin, presented on the vegetable oil. In a pilot reactor operating with the gummed soybean oil, the pressure drop of the reactor increased after some hours of operation, causing interruption of the test and removal of the catalysts and inerts that were analyzed by solid-state NMR. Phosphorus 31 magic wand spinning of the inert has no signal. Phosphorus 31 magic wand spinning of the spent catalyst and the virgin catalyst have no signal from 0 to 10 ppm, but phosphorus 31 magic wand spinning of the inert plus catalyst at the clotted region shows NMR signal from 0 to 10 ppm related to monomeric orthophosphate species due to the phospholipids. By the convolution of the phosphorus spectrum, it is possible to obtain the amount of each species in the samples studied. NMR results led to the conclusion that the phospholipids originally present in the soybean oil decompose at the entrance of the hydrotreating reactor with the rise of the temperature, forming monomeric phosphate species and then polyphosphates. This material accumulates on the inert sand or alumina inside the reactor, leading to the obstruction of the bed and increasing the reactor pressure drop. These observations do not recommend the use of the gummed soybean oil as a feedstock for green diesel production using this process. The biopolymers that constitute the major components of lignocellulosic biomass materials, cellulose, hemicellulose, and lignin, can be used as a precursors in the synthesis of materials with several technological applications and are considered important source of clean and renewable energy. For example, for the second generation bioethanol production, various physical and chemical processes can be used to extract these components from biomass, but an adequate characterization of the lignocellulosic material is not always easily feasible. In this work, with collaboration with the Professor Jair Checon's group at Federal Institute of Espírito Santo in Brazil, we developed new methodologies based on carbon-13 magic wand spinning with cross-polarization for the determination of the main lignocellulosic components and structural changes in these components due to some process. We worked with lignocellulosic residues like sugarcane bagasse, sugarcane straw, sorghum biomass. We use calibration curves, spectral deconvolution of known samples. And finally, in this slide, we can see that in the expanded view of the C13 CPMS, showing the lignin region, and in the table, that the amount of lignin and the structure of the lignin in the studied samples show large scattering. For example, from this carbon-14 NMR spectrum of the sugarcane bagasse after hydrothermal treatment, we can see changes in the lignin structure as well in the lignin content, possibly due to partial removal of carbohydrates and partial lignin changes, partial depolymerization of the lignin. Hydroxyphenyl, syringyl, thiacil are the main building blocks of the lignin structure. Here we can see the SAMPS NMR team, old members, not so old members, a new member, brand new members, a former member, and thank you for your attention.
Thank you, Luis. And let me ask, are there questions?
Yes, methanol is a regular one. And about the phosphorus, I didn't understand it very well, sorry.
Yep. Yeah, here, why don't you ask?
I'm just curious if the phosphorus signals are coming from a ligand-like behavior or it's part of the main catalyst, or the phosphorus catalyst.
Some signals are from the catalyst, but the phospholipids decompose and they produce other species. Then in the phosphorus 13 NMR, we can see phosphorus from the phospholipids decomposition and phosphorus from the catalyst. But some phosphorus from the phospholipids can be distinguished from the phosphorus from the catalyst, can be distinguished very well.
Okay, are there other questions? Yes.
Hi, great talk. So you did show the calorific value as well as like the oxidative amount or whatever, but were you able to get like cetane numbers on any of these biodiesels? There's a fair bit of literature about getting that via NMR. That's pretty easy. Just to like test the quality of the fuel mixes you were creating.
Oh, okay. If I understood well, it is possible to determine the cetane number by NMR means. Yes, I can try. If you send me some information, I can try and answer you.
Okay. Yeah.
Okay, well, thank you, Luis. And
Okay.